Okay, so this is the answers for the Double Award Biology Higher Tier 2019 paper. So the first question states what's meant by the following term, the term gene. So the gene is a section of DNA, and an allele is a different form of the same gene, so that means it can either be big B, big B, big B, little b, or little b, little b. Cystic fibrosis is an inherited condition. The allele that causes this condition is recessive. That's going to be important for the question. The family tree below shows how cystic fibrosis is passed on in families. Use the letters N and N to complete the Punnett square to show how parents 1 and 2 can produce offspring number 4. So offspring number 4 has cystic fibrosis, which we can see. So if it's a recessive condition, like I said in the question, they need to have 2 little n's, which then means we get half our DNA from mum and half from dad. So mum must have a little n, and so must dad, but they are unaffected, so they need to have at least 1 big N. So that's why both parents then here have got big N, little n, big N, little n, and then they're able to produce child number 4 because we get a child who is homozygous recessive. Say the genotype of individual 9, so it's little n, because they need 2 recessive alleles to have cystic fibrosis. Say the possible genotypes of individual 5, so individual 5 sibling of the 2 parents, so they could be big N, homozygous dominant, because they haven't got cystic fibrosis. However, they could also be a carrier and not have cystic fibrosis. Our understanding of how characteristics are inherited comes from the principles proposed by Gregor Mendel in the 19th century. In his experiments of pea plants, Mendel studied characteristics controlled by a single gene, stating why this is not representative in most phenotypes, and it's because it's controlled by multiple genes. The diagram shows a bacterial cell. This is one thing that they can ask from year 10 to remember. So our outside wall is our cell wall, our inside is our cell membrane, and everything in the middle then is our cytoplasm, and it holds the DNA. Caris wants to investigate the effect of an antibiotic on bacterial growth. She set up the experiment shown below with test tubes containing increasing concentrations of an antibiotic and inoculated each test tube with a fixed volume of nutrient broth containing known concentration of bacteria. The tubes are incubated at 25 degrees for 2 days. The cloudier the solution becomes, the more bacteria present. A clear sample means there's no bacterial growth. Explain the result observed in tubes 4 to 6. So 4 to 6, the solution's clear, so all it means is that there's no bacterial growth as all the bacteria have been killed. Suggest the minimum concentration of antibiotic that would be effective against the bacterium, and it would be 1.5. Suggest how Caris could improve her experiment so that she could obtain more accurate value for the minimum concentration of antibiotics needed to be effective. And we'd have to test smaller intervals, because we know at some point between 1 and 2, the solution goes clear where there's no bacterial growth. So if we had test tubes with concentrations of 1.2, 1.4, 6, 8, we can be more specific and more accurate. The diagram shows the 2 types of cell division occurring in a cat in the process of fertilization. The number of chromosomes of a body from the ovary of a cat is also shown. So I'm going to highlight ovary because we're looking now at the sexual organs. So we're looking at certain cell divisions. So state the type of cell division occurring at the points P and Q. So P, we are looking at, we've got our cell, our original cell here, and we end up with 4 daughter cells that are produced. So P must be meiosis. At Q then, we have our original cell here, and we produce only 2 daughter cells, so it has to be mitosis. Say the number of chromosomes in the following cells. So the following cell in A, we've got the sperm cell from a cat, and if we know that the ovary of a cat has 38, in meiosis, the gametes contain half the number, so A needs to have 19. B, the egg cell from a female cat, also has to have 19, because when the sperm and the egg cell combine in fertilization, we're going to end up with 38 chromosomes. C is going to be 38, and then D is 38, because in mitosis, we have identical daughter cells to the original cell. Apart from chromosome number, state one other difference between the daughter cells produced by the cell P and Q. Daughter cells, as a result of meiosis, are genetically different. The daughter cells in mitosis are genetically identical. Rees wanted to investigate the abundance of distribution of 4 plant species as he moved from the edge of the woodland towards the centre. He laid down a tape measure to form a transect. Using 0.25 metre squared quadrat, he sampled every 5 metres along the transect line, estimating the percentage area cover for each species. The diagram shows the area of woodland that was investigated. Draw a line graph of the results of both dogs, Mercury and Bluebell, on the grid below, labelling each line. Okay, so there's 4 marks. So the distance along the transect along the bottom, at the side is the number of each species, and then just plot in the points for dogs, Mercury, and for Bluebell. Take the term used to refer to competition between 2 different species, that is inter-specific competition. Describe the results for dogs, Mercury and Bluebell. Suggest an explanation for your answer. So as distance increases, the Bluebell percentage also increases. As distance increases, the dog's Mercury percentage decreases, so it's the opposite. So what it's telling us is that the Bluebell does not need a lot of light, because as it's going, as the distance into the woods increases, the light intensity would be decreasing because of the coverage from the trees, whereas dog's Mercury needs light. Jim suggested to Rees that he should have carried out random sampling. State why it would be unsuitable to use random sampling method in this investigation is because we're looking at the effect of the environment on distribution. Suggest what improvement Rees could make to his method. We could take samples at more frequent intervals. Biodiversity is a measure of the variety of different species and the number of individuals within those species in an area. So 2 reasons why it's important that biodiversity is maintained. And it's maintained to provide nutrients and food, for industrial materials being produced, for medicines, and for human well-being. They'd love that for this question. Thalidomide is a medical drug that caused unexpected damage to fetuses in the 1950s and 1960s. It was used for easing morning sickness in pregnant women, although it had not been fully tested. As a result of the damage to fetuses, the drug was banned. Drug testing was also made more thorough. Thalidomide has since been used as a treatment for bone cancer. However, its use is heavily regulated to prevent a repeat of the problems it caused last century. With thalidomide came the widespread recognition that differences in sensitivity to drugs between species required consideration. Many scientists report that animals were good predictors of how humans would respond to drugs, and that the current testing procedures would have identified the dangers of thalidomide and the side effects that would have been avoided. Despite this, there's evidence that animal tests cannot predict how humans will respond to a drug. In 1990, 6 different drugs were trialled in humans and in animals. The test results showed that animals and humans had 22 common side effects. There were 48 effects in animals which did not occur in humans, and there were also 20 side effects in humans that did not occur in animals. The sensitivity of the animal test is calculated using the formula. Sensitivity is true positives divided by true positives plus false negatives times 100. Calculate the sensitivity of the animal test. So the true positives are 22, which we've got here in the question, and then it's got 22, and the false negatives is the effects is 20, so it's where I've got them from, times 100, which gives me an answer of 52.38. Use only the information in the passage to answer the following questions. Evaluate the arguments for and against the use of animals in drug testing. So the arguments for is that they can test for side effects. And the reason against is that animals may not respond the same way as humans because we have a lot of false negatives. State how thalidomide problem has one positive outcome on the development of new drugs. It's said in the information that there's now more thorough drug testing. State why the ban on thalidomide was removed and because it treats bone cancer. Negative feedback mechanisms maintain optimum conditions inside the body. State what is meant by the term negative feedback. It's a change from the normal conditions resulting in the body restoring balance. State 3 factors that are controlled within the body to maintain optimum conditions. We need our temperature to be the same, our pH and our blood glucose. Hypoglycemia is a condition which occurs when blood glucose levels fall to a dangerously low concentration. The normal blood glucose range between 4 and 7.5. Suggest 2 reasons that could cause the blood glucose in a person with insulin-dependent diabetes to fall below. They could exercise too much, or they could inject too much insulin, which is going to cause the blood sugars to decrease. Explain why a glucagon injection may be used to treat severe hypoglycemia, because glucagon will travel to the liver, where it's going to convert our excess glycogen into glucose, and then the glucose level will be released into the blood, and the level then should increase above 4 and into normal range. Warfarin is an anticoagulant and has been used as a poison to control the rat population since the 1950s. Rats die from the internal bleeding a few days after eating poison bait. In 1960, warfarin-resistant rats were found in Montgomeryshire. In the years that followed, students found that the area where rat resistant to warfarin were detected was expanding at a rate of approximately 5 kilometres a year. Use your knowledge in natural selection to explain how resistance to warfarin developed and spread. So remember, these are the 6 sentences you need to learn, and you only have to change certain things. I'm going to highlight the bits that I've changed from our 6 sentences that we've learned. So you've got all species of rats, because they could change the animal, show variation. Some have warfarin resistance, I've got that from the question, and some do not. The ones with the warfarin resistance survive, and the ones without die. This is known as natural selection. The ones with warfarin resistance breed and reproduce, passing on the warfarin resistant gene to offspring. This continued over many generations until a new species form. Okay, so our 6 sentence, and we can apply this to any evolution question. We are just changing those 4 or 5 things. DNA is made from 2 long chains of alternating molecules of sugar and phosphate connected by bases. The chains are twisted to form a double helix as shown in the diagram. A sample of DNA was analysed and found to contain 27% thymine. Name the complementary base to thymine. Is adenine. So adenine binds to thymine, cytosine binds with guanine. Calculate the percentage of cytosine that will be present in the sample. So if we know that 27% is thymine, and we know that every thymine binds with adenine, that we're also going to have 27% adenine, which in total will give me 54% of the bases are adenine and thymine together. If I take that from the 100%, I have 46%. And that 46% is the total of cytosine and guanine together, but we're only looking at cytosine. So then I do the 46% divided by 2, so I've got 2 more bases, which gives me 23%. So 23% is cytosine, which also then means 23% would be guanine. Explain how the sequence of bases of DNA determines the structure of a protein. Well, we have the triplet code, and that is saying that 3 bases will code for a specific amino acid, and that the amino acids will make proteins when they join together. The sequence of bases on a small section of DNA is shown below. So we've got TGA, CTG, GTC, AAC. This section of DNA can undergo 2 different mutations. Mutation 1, where we've got a change in the second base, and mutation 2, we've got a change in the last base and that's it. Use information and your own knowledge to conclude why mutation 1 may affect the organism more than mutation 2. So in mutation 2, 2 triplet bases have altered. So in mutation 1, we've got the second base and the third base has had altered codes. Mutation 2 only has 1 triplet base that is altered, and the altering bases will alter the amino acids that is going to be collected, so therefore a different protein may be produced.